webinar series dedicated to open innovation in lymphoid neoplasm, organized by the Carnot-Kalim Institute. Our goal is to bring together experts from academia and industry to discuss the latest breakthrough and challenge in the field. The Carnot-Kalim Institute is a French research consortium specialized in lymphoid malignancy. It unites top-tier academic laboratories and clinical research centers to accelerate innovation and improve patient outcome. And today's episode will explore diffuse large B-cell lymphoma and high-grade lymphoma, focusing on the different types of MYC rearrangement and their implication for biology, prognosis, diagnosis, and treatment. And for this Red Talk, we have a prestigious guest, David Scott. I have to stress that it is the last episode of the season, and we will be back in 2026. Now, Thierry and Clémentine will present our guest, David.
So it's our great pleasure to welcome a colleague and friend, Dr. David Scott, that is the Deputy Head of the Department of Lymphoid Cancer Research at the University of British Columbia and Clinical Director of the Center for Lymphoid Cancer in Vancouver. Dr. Scott is an internationally recognized expert in the field of lymphoma genomics and translational research. His work has contributed to our understanding of how genetic alterations, including MYC rearrangements, influence disease behavior and patient outcomes. David is also a fantastic mentor and teacher, and today he will guide us through the biological and clinical impact of these rearrangements and how they shape modern diagnostic and therapeutic strategy. So please, David, the floor is yours.
Thank you very much, Clémentine, for that introduction. Thank you very much for inviting me, or the organizers for inviting me, to give this talk, which is the finale of season 5. So my topic is talking about different MYC rearrangements in DLBCL and high-grade B-cell lymphoma. And I'm going to be giving quite a compact talk to allow for lots of questions, and hopefully I'll be able to answer some of them. So here are my disclosures, and I think the important ones to note are that I consult for a company, or have consulted for a company called Arima Genomics, who is working in the space, and that I also have patents which are involved in gene expression to identify subtypes of aggressive B-cell lymphoma. So this talk is going to introduce or summarize some of the work that has gone on at BC Cancer, but it rests on a foundation that goes back over 50 years. And I appreciate that there are lots of people, including people who are in the audience today, who have contributed a lot to this particular area. And I'm not going to be able to cover everyone's contribution. So I apologize for that in advance. And I do encourage also people to go back, like I did on the weekend, to YouTube and look at Dr. Rainer Siebert's talk on a related topic from 2 years ago. So I'm going to start with 1 minute on history. I'm then going to talk about how MYC rearrangements feature in the current classifications before going on and introducing the incidence and architecture of MYC rearrangements across the range of aggressive B-cell lymphomas, touching on what we think is the etiology of these rearrangements and why the rearrangements are different in high-grade B-cell lymphoma with MYC and BCL2 rearrangements, and then address the topic of, are we just defining things by MYC rearrangement? Are these identified groups actually biologically coherent? And do they describe the core of something greater? And then finally, I'm going to talk about practical considerations, addressing issues with FISH, and spend just 1 minute at the end on treatment, which is increasingly controversial. So going right back to now almost 50 years ago, looking at Burkitt lymphoma, Zek and colleagues at the Karolinska first described the T14 rearrangement, shown with these arrows here, with material from chromosome 8 ending up on chromosome 14. And then 6 years later, Riccardo Della Favara in Carla Croce's lab in New York described that this rearrangement actually involved the MYC oncogene. I think it was, and I'm going to be corrected on this, in the early 90s that what we now call double hit was first described in a paper first authored by Daphne de Jong in a case of transformed lymphoma. So how do our aggressive B-cell lymphoma classifications look today? And obviously this is very, very simplified. On the left, you have the international consensus classification. On the right, you have the WHO Hem5. We start off with morphology, and then MYC rearrangement testing is encouraged across the board, essentially, of aggressive B-cell lymphoma. And then we have categories such as Burkitt, which requires MYC-IG rearrangement, and you've got these high-grade B-cell lymphoma with MYC and BCL2 rearrangements, which also require a MYC rearrangement to be present, in this case with a BCL2 rearrangement. And also, it must be absent in high-grade B-cell lymphoma with 11q aberration. Now, luckily for us, the classifications are largely concordant, with the exception of aggressive tumors with MYC and BCL6 rearrangement, which has been kept as a provisional entity in the international consensus classification to encourage additional study and testing, whereas it's been removed or relegated to the WHO Hem5 in such a way that if you have a MYC and BCL6 rearrangement, you end up in either high-grade B-cell lymphoma NOS or diffuse large B-cell lymphoma NOS, depending on the morphology of the malignant cells. And I'm pleased that we are aiming to move towards one classification again in 2026, and there should be a draft of that classification later in 2026. So what are the incidence of MYC rearrangements as determined by FISH across the aggressive B-cell lymphoma entities? Well, obviously it's required for Burkitt, so that's 100%, and also for high-grade B-cell lymphoma with MYC and BCL2 and MYC and BCL6 rearrangements. It's then also seen in about 60% of plasmablastic lymphomas, just under 50% of high-grade B-cell lymphoma NOS, and in the range of 3% or so in tumors that we call diffuse large B-cell lymphoma NOS. Now, to put this into context, this is the percentage of aggressive B-cell lymphomas. So these entities that we're defining or have high incidence of MYC rearrangements are making up a small proportion of what we see in the clinic, where the vast majority of our aggressive B-cell lymphomas continue to be in this diffuse large B-cell lymphoma, not otherwise specified category. There has been a lot of work over the years looking at breakpoint architecture and partners. And what I'm going to present here today is the work of a survey that was done with the LLMPP and the Burkitt Lymphoma Genome Study Project, and first authored by Laura Hilton in my lab, where we looked at breakpoint architecture as determined by either whole genome sequencing or capture sequencing of the MYC region in association with common partners. And I'm going to go through these one at a time. What you're going to see here, though, is what we call a Circos plot, where the chromosome segments are shown on the outside. We have the genes in the inner ring, followed by a ring here, which shows the different pile-up of mutations across all of these cases. And a lot of these mutations are related to AID activity. And then each of these lines go from chromosome 8 across to the partner gene. So each one of these lines represents an individual tumor. So starting off with Burkitt lymphoma, which is not admittedly the specific topic of the talk today, these rearrangements are exclusively to IGH, IG kappa, and IG lambda, with IGH making up 80% of cases. And there's essentially, well, there can't be any non-IG partners seen. If we then go across to those tumors that have what we call a sole MYC rearrangement, so high-grade B-cell lymphoma and diffuse large B-cell lymphoma NOS, most of these are also to immunoglobulin partners, predominantly IGH, but also a small amount of kappa and lambda. And then there are some rare other partners, including PAX5, Raftlin, and LPP, which are all genes that are active and expressed in the germinal center. You also have here MYC with BCL6 rearrangements, and these largely look like sole MYC rearrangement patterns, with the exception of what we call pseudo double hit, which is where there's been a single rearrangement between MYC and BCL6, the T3H shown here. And I just wanted to highlight some work by Russell Ryan almost 10 years ago now that showed that in that particular rearrangement, BCL6 wasn't driven. So those rearrangements act as if they are a sole MYC rearrangement. And then you have the exception, which is MYC and BCL2 double hit. And what we see here is that 50% of these rearrangements are with non-IG partners, and there is definitely heavy recurrence here with PAX5, BCL6 LPP, and then less so with Raftlin and with IRAG2. And you can see also that you have a greater proportion here of IG lambda and kappa, and collectively they're almost at the same level as IGH. And that's important when we think about FISH strategies. Now, this is a little bit of a generalization, but the patterns we see when we hone in on the IGH locus, where there's been an IGH MYC rearrangement, we can see that the rearrangements fall within class switch donor and acceptor sites, with roughly equal numbers, or maybe a predominance of donor sites here with EMU and Burkitt lymphoma, more evenly spread between donor and acceptor in DLBCL NOS. And I do want to highlight that there is this grouping here, which are called V, and these are on the border of the V and EMU, and in some cases within the V, and these look like they are non-class switch or driven by non-class switch AID activity. Now, the other interesting pattern to see here, and this is important to think about how the MYC is actually driven, is that the breakpoints are in IGH MYC rearrangements in intron 1 and upstream of the gene. And the question is, why do we see that pattern? So this is a cartoon of slight class switch recombination on chromosome 14. You have AID-induced breaks in EMU and in a downstream acceptor site. That segment of DNA is then looped out and circularized and eliminated, and you then get the rearrangement which produces class switch. We speculate through the patterns that we see that, for example, we have a break in the donor site, in the acceptor site in this particular case, and an intron 1 in this instance of MYC, and then we produce a derivative 14 where the MYC is on the same derivative chromosome as the RR enhancer element, which then drives expression. And this could also be seen if we use the donor site in EMU, we would get a similar rearrangement where the MYC gene is on the same derivative chromosome as, in this case, two RR elements that then drive expression. So this really results in enhancer hijacking, and if the breaks were actually downstream of MYC, then the gene would actually be separated from these enhancers, and the expression would not be driven. Now, the breakpoints in MYC for IG kappa or IG lambda and almost all of the non-IG rearrangements are typically downstream of MYC, and you can see this sort of forest of rearrangements. And these are hypothesized to occur through AID activity, but not class switching. The partner genes are typically highly expressed in B-cell differentiation, particularly the germinal center. And the breakpoints are related to maintaining the partner gene enhancer on the same derivative chromosome as the MYC gene, explaining why they're downstream. And importantly, when we think about strategies to detect these breakpoint regions on chromosome 8 are spread over about 1 megabase, so quite a large region. So why is it important to, why is this important to the B-cell lymphoma? Well, MYC is very tightly regulated in B-cell differentiation. And I think that when people first developed MYC immunohistochemistry and applied it to lymph nodes, they expected broad swathes of cells to light up and to be expressing MYC. But in fact, when this was looked at in two really seminal papers in 2012, they saw that these cells were extremely rare and tended to be in the light zone, but also were important for initiation and initial formation of germinal centers. So we think that we get a burst of MYC as an important event in entry and establishment of the dark zone in the germinal center, but also we have a burst of MYC as cells exit or re-enter the dark zone. And this activity on re-entry to the dark zone is very rapidly squashed, particularly by BCL6 expression. And so we think that MYC is really acting as a transcriptional amplifier and is important to initiate that re-entry, but then other events take over to drive different proliferation and cell division, particularly cyclin D3. So why or how are the MYC rearrangements in MYC and BCL2 double hit different? Well, first, 50% of the MYC partners are non-immunoglobulin. And this is not quite exclusive to this entity, but is heavily enriched with 50%. And this was first noted in a very important paper from the LYSA group led by Dr. Kobe Bergman, and then was expanded upon in a paper from the LLBC led by Andreas Rosenwald. What we also see is this push towards these breaks in IGH being almost exclusively within class switch acceptor sites, as opposed to Burkitt and the others, where it's either favored towards EMU or essentially exclusively EMU. So why are they different? Well, they're different, or we think that they're different, because they're occurring in cells that have a pre-existing IGH-BCL2 rearrangement, which occurs through off-target RAG activity in the bone marrow. We know that MYC expression drives apoptosis, and so it would be advantageous for these cells to preserve BCL2 expression to fend off the apoptosis. And we know that most, if not all, B-cell lymphomas, they may not rely on B-cell receptor expression, but they appear to rely on IGH expression. So what we see here is this is the T1418 precursor, where you have chromosome 8 here with MYC. You have your intact chromosome 14, and then you have this derivative 14 chromosome from the 1418 translocation. So in this scenario, you could have, first of all, you could have non-IG partnership, which would preserve both the T1418 and also the intact chromosome 14, or you could attack these chromosome 14 or derivative chromosome 14, but you have to do it in a very specific way that fits with the patterns we see. So this is one way. You have a derivative 14, which has the MYC gene, which is still driven by the RR element, but because the donor site the rearrangement is in the class switch acceptor site of the derivative chromosome, BCL2 is still potentially driven by EMU or in fact an RR element. So that's one pattern. The other pattern is that it actually attacks the intact chromosome 14, again going into this acceptor site, and so your IGH can still be driven by EMU or downstream elements. What doesn't work to preserve both BCL2 and IGH is to actually go into the EMU the donor site of either the derivative chromosome, because now BCL2 isn't driven, or of the intact chromosome 14, because now IGH is not driven. So the way that these lymphomas still get MYC constitutive expression and hijack these enhancers is either to use non-IG elements or to go into the acceptor site. Non-IG partnership, we have arguments about whether in fact it's important, and one piece of evidence that it's functional is that there is elevated MYC mRNA expression, and what's shown here is RNA-seq data with normalized MYC expression in Burkitt, DLBCL, NOS, and high-grade B-cell lymphoma with MYC and BCL2 rearrangements. This dotted green line here is the mean expression in Burkitt lymphoma of IGH rearranged cases, and the gray line is DLBCL that does not have a MYC rearrangement. And you can see that in Burkitt, IGH is associated, partnership is associated with slightly higher expression than both IG kappa and lambda. It seems to be fairly equal when you look at double hit lymphoma, and you can see that both PAX5 and BCL6, which are heavily recurrent partners, and also other non-IG partners, are both expressing a higher level of MYC than non-rearranged DLBCL, but it is significantly lower than what we see with immunoglobulin. So we think it's important, but it does explain potentially differences that we see in prognosis, which I'll tell you about later. So one question that we were confronted with in 2018 was, are we defining things through rearrangements and lumping them together based on this without any other shared biology, or are we identifying something that is biologically coherent and shares biology? And I think that it wasn't really a surprise to everyone that this entity of Burkitt, which was described, middle of last century, and has a very nicely defined morphology and requires requires IG partnership, was actually quite homogeneous on a mutational level. Then the question was, what about MYC and BCL2? And there were papers in the late 2000s which suggested that maybe these weren't actually a coherent biological entity. So we looked at gene expression profiling, and this is now an oncoplot of whole exome and whole genome sequencing for just over 200 cases. And we can see a few things. First is that the most recurrently mutated genes are shared with follicular lymphoma, which we think suggests a common precursor cell population, or that some cases are arising from occult follicular lymphoma. We saw that almost all of them belong to the GCB subtype of diffuse large B-cell lymphoma. They express something called the dark zone signature, and they're over 95% belong to the EZB genetic subgroup as defined by Lou Stout as the lymphgen. And then there are also shared mutations with Burkitt lymphoma, such as MYC, FOXO1, cyclin D3, and DDX3X. And these are genes that regulate the dark zone of the germinal center. So the other thing that both Burkitt and double hit MYC and BCL2 share are that the cells at a gene expression level look like germinal center B cells from the dark zone. So this work goes back to 2006, and this was introduced in this very nice webinar in this forum by Rainer Siebert, and this was the description of molecular Burkitt, which was a comparison that arose from Burkitt lymphoma versus diffuse large B-cell lymphoma. And they defined a signature which was consistent with Burkitt, but also drew in a number of other cases that were not called Burkitt. And this has been distilled down to a signature called the molecular high-grade signature by the Leeds group. The second piece of data that supports this notion was the work from Gabriel Victora, working with Ricardo Della Favara, where they looked at the difference between physiological dark zone and light zone B cells and produced an interesting signature from that. And then we produced something called the double hit signature, which was comparing MYC and BCL2 double hits versus the parent population of GCB DLBCL. And we have subsequently renamed that the dark zone signature because it is positive and strongly positive in all Burkitt lymphoma. So this sort of converges to this idea of dark zone lymphoma, which is an umbrella term that encompasses a number of entities, including Burkitt and double hit lymphoma. So these lymphomas encapsulate all Burkitt, 80 to 85% of MYC and BCL2 double hit cases, but also scoop in high-grade B-cell lymphoma NOS and a proportion of GCB DLBCL NOS. And I'm not going to talk much more about that because it's a little bit tangential to the topic today. But one interesting thing is, what about these double hit MYC and BCL2s that don't express this, what we think is a summative gene expression profile? And I have to say the numbers are pretty small, so we can't draw firm conclusions, but they tend to have an atypical immunophenotype, and a proportion of them are CD10 negative, which is typically strongly positive in both Burkitt and in double hit lymphoma. There's a trend towards more non-recurrent MYC partners and cases where we can't identify the partner with our technologies, and they certainly have lower MYC and BCL2 mRNA levels using RNA-seq, but also our nanostring assay. And again, very small numbers, but when we treat these patients with R-CHOP, this is a retrospective analysis, we can see that their outcomes are very similar to diffuse large B-cell lymphoma NOS. Then the question is, what about MYC and BCL6 double hits? And this is obviously, again, relatively small numbers, but we see much more heterogeneity in both the mutational landscape, but also the cell of origin, the expression of that dark zone signature, and the lymph-gen subtypes that they fall into. So where do we sit with double hit lymphoma in 2025? I think that summing all of this up, I favor what's been put forward by the WHO Hem5, where these MYC and BCL6 rearrangement cases are pushed back into DLBCL NOS or high-grade B-cell lymphoma NOS. So then what about those cases that have a MYC rearrangement, but it's a sole rearrangement, and they don't have typical Burkitt morphology or immunophenotype? So something stopping us from calling them Burkitt. These make up, as I said before, about 50% of high-grade B-cell lymphoma NOS, and a much smaller proportion of DLBCL NOS, where they're split between GCB and ABC. And there aren't a huge number of these to look at, and we've focused our sequencing efforts on those cases that demonstrate the dark zone signature, which make up about 40% of these cases. And when we look at these, the mutational landscape of these cases and compare it to Burkitt lymphoma, this landscape is actually very similar. And when we apply a random forest model, which was developed by Nicole Thomas in Ryan Moran's lab, we can see that 50% of these cases would be called Burkitt lymphoma based on their mutational profile. So this sort of leads to this question, bit provocative from a pathology point of view, is, are these dark zone MYC rearranged, particularly high-grade B-cell lymphoma NOS, but to a certain extent DLBCL NOS cases, being misdiagnosed, and they are in fact Burkitt lymphoma? And that, I think, will hopefully incite quite a lot of discussion after my talk. Now, all of this information has practical considerations. I think that most of us are identifying MYC rearrangements using FISH probes, and the recommendation is that based on the spectrum of MYC rearrangements, you should start off with widely spaced break-apart probes, particularly as there are a number of partners reducing the utility in a screening sense of using dual fusion probes. So you can see that if you put probes very close to MYC here, you're going to get most of your IGH rearrangements, but you're going to miss almost all of your IG kappa and lambda rearrangements, which we think are important, even in Burkitt, but also all of your non-IG rearrangements. Now, in this process of looking at the dark zone signature, we asked the question of why aren't all of these cases double hit? And when we looked and did whole genome sequencing, we saw a phenomenon which we call cryptic rearrangements. And this is one example here of MYC, where there's been 2 breaks in chromosome 8, and the PAX5 super enhancer has been inserted here. So you can see that the red and green signals are still intact, so no break apart is seen. And this is a phenomenon that was seen in the same year by Rainer Siebert's group in a pediatric case of Burkitt lymphoma. Now, dual fusion probes can detect some of these rearrangements, and there's a very nice large series from Becky King and Susan McPhail at the Mayo, where you can increase your detection rate somewhat, and they propose using both break apart and then MYC IGH dual fusion probes. This will increase your rate by 1 to 4%, but it will still continue to miss IG Kappa Lambda and non-IG partners. So you then would have to default into using a large suite of dual fusion probes, which most laboratories aren't particularly keen on doing. Finally, there is the very confusing phenomenon, which has created a lot of concern, which is this unbalanced FISH pattern, where you, instead of getting a break apart, you actually lose one of the signals, either the red or the green. And the question has been whether the MYC gene is deleted and whether a rearrangement has occurred. And a very nice survey, again run from the Mayo, demonstrated that 50 to 60% of reporting labs in North America really said, we don't know what this means, and pushed that back to the clinician to interpret. And about 5 to 10% of labs actually said that there was no rearrangement present. So we looked at this. We saw that this unbalanced pattern, very similar to the Mayo results, was present in 13% of MYC and BCL2 double hit tumors. And we could show that the architecture, the rearrangement of the architecture, actually preserves the derivative chromosome that contains MYC. And I think in the absence of additional information, these should be reported as being positive for MYC rearrangement. Now, treatment of MYC and BCL2 double hits is controversial. In North America, we followed a gathered cohort, or large parts of North America, we followed a gathered cohort from Petrich et al. In 2014, which showed that outcomes with R-CHOP were very poor, whereas with escalation, they were significantly improved. And we put in a policy in place in 2014 that in patients under the age of 75 who were fit, they should be escalated to dose-adjusted EPOCH-R. There's never going to be a direct randomized clinical trial comparing the dose-adjusted EPOCH and R-CHOP, so we decided to do an era-on-era comparison where FISH was done on all DLBCL tumors of patients diagnosed across the province of British Columbia. So we have a historic cohort, which the curves are shown in red here. This was where FISH was done retrospectively in cases between 2005 and 2010, and R-CHOP was the guideline. And then in 2015 to 2020, where FISH was done on all cases in the clinical lab, and the guideline was to receive dose-adjusted EPOCH-R. Now, this is an era-on-era comparison. So the DA EPOCH era includes a number of patients who did not get dose-adjusted EPOCH for various reasons. I consider this a practice-affirming study. We did see a significant improvement both in freedom from progression and also overall survival. But again, this has a retrospective nature to it and has those caveats there. So practice-affirming, we feel comfortable continuing on with our practice of dose-adjusted EPOCH. And this is very in line with another study from the Flatiron group in North America, which was a gathered cohort. There's been a lot of discussion about what about the prognostic significance of non-IG partnership. This is the largest study to date from the LLBC. Largely, these patients were treated with R-CHOP, and it brought together clinical trials and real-world experience. Down here is the IG-partnered double hit cases. Up here is the double hit with non-IG partner, and you can see that that's significantly different, with non-IG having better outcome when treated with R-CHOP or similar. Very small numbers, but we've also looked at this in our historic comparison. This red line here is the patients who were treated in the historic era with IG partnership, and this blue line is in the dose-adjusted epoch partnership era. And so we think that this benefit of dose-adjusted epoch is certainly more evident in IG versus non-IG MYC partnership cases. Intensification has been an important part of treatment for Burkitt lymphoma for some time, and I've shown you the data in double hit. We have absolutely no data to inform on whether intensification works for other dark zone lymphomas, including those with sole MYC rearrangement. But that is the strategy that we're going to be adopting in British Columbia going forward, starting next year. So to conclude, MYC rearrangements occur across actually a broad range of aggressive B-cell lymphomas and form part of the definition of both Burkitt lymphoma and high-grade B-cell lymphoma with MYC and BCL2 rearrangement. The MYC rearrangements that we observe are consistent with their origin involving AID activity, particularly class switch, but also other activities in non-IG, IG, kappa, and lambda, and the consequence being dysregulation of normally very tightly controlled MYC expression. The pre-existing T14-18 translocation in the development of double hit leads to a different pattern of these rearrangements, favoring non-IGH partners, and when IGH partnership is used, this occurs into class switch acceptor sites. My leaning on this is that Burkitt lymphoma and double hit have both defining in and of themselves and shared biology that go beyond the MYC rearrangement, and they form the core of what we're calling dark zone lymphomas. And this dark zone lymphoma group includes additional MYC rearranged tumors that look like Burkitt on a mutational level. And finally, the range of MYC rearrangement architecture has an impact on detection strategies when using FISH, prognosis, and also possibly management. So I'd like to end with an acknowledgement slide, particularly Laura Hilton, Brett Collins, Walid Al-Duwage, and Susanna Ben Naraya from our group, but also Ryan Moran, Lisa Imzalou-Stout, and the LLMPP, and my clinical and pathology colleagues at British Columbia. And thank you for your attention.
Thank you very much, Dr. Scott, for this insightful presentation. And we'll now open the floor to questions. So please use the Q&A section to submit yours, and we'll share them with our speakers. So maybe Fabrice, you have a question?
Yes, maybe I can start. Thank you, David, for your nice talk. I think you published recently algorithm to identify dark zone lymphoma with immunohistochemistry. Can you give us more details regarding this publication?
I can. So this publication was led by Eric C. At his time when he was in Wake Forest and now at the Mayo. And this algorithm was developed by him and his colleague, particularly starting from the standpoint of what genes and proteins are highly expressed in the dark zone versus the light zone of the germinal center. And then the question which started that off was, can we differentiate Burkitt from diffuse large B-cell lymphoma? So this algorithm includes 5 genes, MYC, AID, STAT3, GC-SAM, which is LGALS, and also SEMA7A. And I think that it does a very good job of identifying Burkitt from DLBCL. I think that it hasn't been tested on a broad enough range of dark zone. So that's now an ongoing project that we have in collaboration with Eric and a number of other pathologists. And in parallel, we're also developing an IHC algorithm that is more based on what we see in the tumors themselves. So we're pulling in a number of other IHC markers. I think that immunohistochemistry is going to be important because it's hard to envisage everyone across the world being able to deploy a gene expression-based assay as part of routine pathology. We have such an assay as part of routine pathology here in British Columbia, which we offer across our province. But I think really widespread adoption of dark zone lymphoma, much like cell of origin, ABC-GCB, will require an immunohistochemistry surrogate.
So currently you use nanostring technology to identify dark zone in your daily practice?
Yeah, we do. We use a clinical grade equivalent of the assay that we call the DLBCL 90. So it has modules for ABC-GCB. It has a module for PMBCL versus DLBCL, and it has the dark zone versus not dark zone, and it reads out from a nanostring platform.
And for the double hit cases, MYC and BCL2 that do not have the dark zone signature, do you still escalate to DA POC in the routine practice?
Yeah, that's a really challenging one, isn't it? At the moment, the gene expression profiling is sort of sitting in the background. As of next year, we're going to hopefully have the ability to escalate all dark zone signature positive patients under the age of 75 and appropriately fit to dose-adjusted EPOC. And then we're going to have to start wrestling with those double hit MYC and BCL2 cases that are not dark zone. Our organization, when you introduce a test, they say that they would like to reduce or limit other tests. And I think that what we're going to be facing, and I think maybe this isn't a bad thing, is that in a year or two, I think we're going to be asked to reduce our FISH usage, and we're going to be restricting MYC and BCL2 testing to tumors that are dark zone or high-grade lymphoma NOS or Burkitt, but we won't be using it across the board for diffuse large B-cell lymphoma NOS. This will create a sort of a split somewhat of our practice from the WHO because we won't necessarily be identifying all of those double hits. But I think that I anyway favor the downstream biology phenotype as the readout rather than just the rearrangements that we can pick up with FISH testing.
Okay, thank you. Just a question, David, concerning the MYC and 11q aberration. As you know, there are a few reports sometimes of cases in which we might find the two. Did you have any experience concerning the presence when you studied of your MYC cases in which there was really the aberration with amplification and deletion? And what is your experience on that?
Yeah, our experience isn't as extensive as Reiner's, Reiner-Siebert's. We do see cases where both occur, and I think that this is a real phenomenon. I don't think that they're completely mutually exclusive. And I think if you look hard across a broad range of aggressive B-cell lymphomas, you see it beyond what was initially described as the molecular Burkitt group. So I think that it is important, and I'm not entirely sure exactly what it's doing, but it's an important original finding. But I think it is challenging to work out how it fits within the classification. I was very pleased when the WHO put out a very prescriptive comment about how or what tumors should be tested. Because if you do look across DLBCL and if you do look across plasmablastic lymphoma, you will see these specific aberrations, and it would be very hard to then work out, what they mean and how to treat those patients.
But it's true, for example, that we have seen a case in which really it was looking like a Burkitt, but in fact, we did the two on emergency and we found the two together. And we were very embarrassed at the end to classify this case. And it was a children, but finally we ended up too difficult to classify with very, it looks like Burkitt, but there was still this 11q aberration. So do you think in your Burkitt series, did you find the 11q aberration in some cases?
I'd have to defer to Ryan and Laura Hilton, but I do think that we did see rare cases.
Rare cases.
So I don't think it's absolutely mutually exclusive. And, you know, I think that there are other factors, obviously, that went into your diagnostic call. But I would strongly favor if they have the correct immunophenotype and the clinical presentation and a MYC IG rearrangement, that they be called Burkitt as a much more sort of defined entity that has been, you know, worked up and defined over decades.
Okay, thank you.
There is a question from Bertrand Nadel. Fantastic talk, David. Thank you. There are new generations of MYC inhibitors, such as OMO-MYC, OMO3103, which are moving to phase 2 in solid tumors. Is it reconsidered in Burkitt lymphoma and other DLBCL or high-grade MYC entities described today?
Yeah, so I haven't seen any studies in that. I mean, I think that it's very interesting to start. I mean, I talked about escalation. I think that that's it's using a hammer in a slightly different way, whereas we really want a scalpel that will go in and attack different parts and different pathways. And we are building up and have built up that sort of knowledge about what pathways are being utilized in Burkitt and, in fact, in dark zone lymphomas, which I think that we can attack. And one very nice thought is to actually attack MYC itself with these inhibitors. And there have been various inhibitors that have been trialed. But I think now we have, you know, degraders and specific inhibitors which are much more targeted than previous agents. The other thing which is very interesting is the use of BCL2 inhibitors. And I think that they have the potential, not in Burkitt, of course, but in MYC and BCL2 double hit. And, you know, it'd be very interesting to go back and look at the translational side of the Cavalli study, for example, to see whether we could identify a group beyond BCL2 expression that benefited. And then we have the Viper regimen, which was published by Wyndham Wilson and Lou Stout, where they hypothesized that if you inhibit the BCL2 expression in double hit lymphomas, that then suddenly you would unleash the pro-apoptotic side of the MYC rearrangement and kill off the tumors. And they saw a 50% overall response in those relapse refractory double hits. So I think we're sort of at, you know, hopefully we're at the beginning of actually using these sorts of designations and entities and descriptions to actually drive precision medicine. But I think that that's going to require actually identifying these tumors beyond, you know, Burkitt and double hit, either retrospectively or prospectively in clinical trials.
Questions from Dr. Corinne Hayoun from Créteil. What is the percentage of transformed lymphoma from follicular lymphoma in the series of in your series of double hit lymphoma?
Yeah, so these are really interesting, and I think that we presumed that when we looked at the genomes and exomes of these tumors that we'd see quite a lot of difference, and in fact on a genetic level we couldn't distinguish them. So transformed from follicular versus apparently de novo MYC and BCL2 double hit looked identical. And, you know, there's a number of, you know, thoughts about how MYC and BCL2 tumors arise. The sort of sharing of those 5 most recurrent mutations, many people have interpreted, say, well, all of these patients actually have occult follicular lymphoma, which is a full-blown, you just don't detect it, and then you get a transforming event. I think there's also the possibility that you have a precursor cell that can spit off follicular lymphoma and can spit off a diffuse large B-cell lymphoma or a double hit lymphoma. And we do have patients where we have diagnostic and relapse samples, or we follow them for many years and we never see the follicular lymphoma. And interestingly, we have double hit cases which relapse late. And when we look, some of them have lost their MYC rearrangement. They have exactly the same BCL2 rearrangement. And there are other cases that are double hit at both time points, but have a brand new MYC rearrangement. So we think that there are these common precursor cell populations that are spitting off these lymphomas over time.
But you did saw some cases with that some double hit relapsing with an FL histology and also clinical behavior.
Sorry, can you repeat that question?
You did have in your cohort some double hit BCL2 MYC that did relapse with only the BCL2 translocation and also with the clinical behavior of a follicular lymphoma. I mean, very indolent, or did they kept the aggressive form?
Yeah, so our series was primarily focused on where we had 2 aggressive lymphomas, but we do have cases where you know, and I think you actually helped put together those cohorts, so you know that we're looking at that. There are cases where we do have aggressive that then comes back as indolent, and they are in many cases clonally related and have the same BCL2 rearrangement, which would suggest, I mean, it may be that they had follicular lymphoma all the way along, or in fact they have a new, you know, a de novo follicular lymphoma that arises afterwards.
Another questions concerning the single hit MYC DLBCL, which looks like a bucket on the molecular point. You cannot find any other. I can assume that maybe some variability of tumor cells in Burkitt and may explain the kind of the potential polymorphism of Burkitt, but you cannot find on the molecular background 2 different kind of potentially Burkitt one and a potentially non-Burkitt. No, it's all the same for you, Burkitt on the molecular point of view. There is ID3, TCF3, and all the molecular features on the NGS, or yeah.
Yeah, so they're not, you know, not identical, and there's heterogeneity both within Burkitt and with these other cases. And what I think what we need to do, and we're in the process of doing, is to actually send all of these cases back to our LLMPP pathology panel and ask them specifically why these cases weren't called Burkitt. Because in some cases, I think it's just some variability or some unacceptable variability in morphology and the cell size and appearance. In other cases, I think it's probably a little bit of BCL2 expression. And in some cases, I think that they look like DLBCL. So it'd be important to sort of tease that out, because I think going forward, you know, if we are going to think about a classification which responds to some of this data, we could think about expanding the definition of Burkitt, which I think would be challenging. Or we could actually say, look, you know, these things belong somewhere. They shouldn't be still left in DLBCL and high-grade B-cell lymphoma NOS. And we'll say that these are a dark zone with a MYC rearrangement, and then try and sort of gather them together and work out exactly how they behave. And I'm sure a lot of other clinical colleagues on this call have had cases where the patient presents like Burkitt. They have a mass in the GI tract. They have a MYC rearrangement, but there's something that stops the pathologist calling it Burkitt. And we go, okay, well, we'll give this patient R-CHOP. And then they briskly tumorize. And we go, oh, that was a surprise. So I think that, you know, there are these sort of clinical behaviors that are distinct from Burkitt or have similarity to Burkitt. And I think we need to explore and understand them. I have to say, you know, we are talking about rare cases. This is not, you know, it's a small proportion of the cases we see over time. But I think that they are going to be therapeutically important.
And they had the dark zone signature, the MYC Sol DLBCL.
Yeah, so these are the ones that these are the ones that I'm talking about. So if you take out the dark zone Sol MYC rearranged DLBCL, the 40%, then the proportion of GCB that have a MYC rearrangement that don't fall into that category is very small. But we do see a number of ABCs, maybe about 3% of ABC have a MYC rearrangement. And I think that is occurring slightly differently in terms of the timing and B-cell differentiation. And I wonder whether that's actually it's acquired in an extrafollicular location. And they typically don't have the dark zone phenotype. There's a really small group, which are an exception to that seem to be very aggressive clinically. They're extraordinarily rare, and we're trying to gather these cases. But those specific dark zone MYC rearranged ABC cases look more like a naive B cell. And sort of probably fit somewhere in the spectrum from BALL and DLBCL. So we're working hard on that, but we're talking about minutia. I think we've got 10 cases across all of our institutions.
Questions from Dr. Nadel concerning the cooperation between EBV and the different MYC entity. Do you have some insights on?
Yeah, so I mean, of course, the biggest thing there is in Burkitt. It was a strong feature of endemic Burkitt, but we also see it in sporadic Burkitt. And it's probably a better way of splitting up Burkitt than pediatric adult or sporadic endemic when we look at the genetic mutations. I think that EBV is a really important way of combating the pro-apoptotic aspects of the MYC rearrangement and is almost certainly an event that precedes the MYC rearrangement. I'd point you to Rainer Siebert's excellent talk from a couple of years ago, which highlights methylation differences and mutational differences between those two. We very rarely, in fact, I can't think of a case, although I'm sure the pathologists on the call will have numerous cases of EBV positive double hit. It does occur, but it's at a much lower frequency. We're talking, 1 or 2% probably. And we haven't really seen it so much in those sole MYC rearranged cases that look like Burkitt. But I really think that that's related to the populations we've looked at, which is in British Columbia, where we exclude PTLD, and we do all sorts of things like that, where that incidence of EBV is significantly higher.
Thank you. There are questions from Dr. Merliot from Bordeaux about MYC rearrangement and double hit in lymphoma of immune privileged sites.
Yeah, so that's an interesting question. We do see double hit in both the testis. We're talking about MYC and BCL2, and we do see it in what we would call primary central nervous system or primary DLBCL of the central nervous system. So it definitely is there, and I think it's probably easier to talk about dark zone than it is about double hit per se. But, when we think about primary CNS lymphoma or we think about secondary CNS lymphoma, that's really made up of ABC, predominantly the MCD subtype, but also dark zone and double hit. So there seems to be a propensity or an ability to go into the central nervous system and proliferate. And we see predominantly a leptomeningeal presentation for the double hits and the dark zones, as opposed to a parenchymal for the ABCs. So I'm not sure I've really answered that question, but it is something that we certainly see.
There is another question from Bertrand with regards to the detection of the cryptic rearrangement.
Yeah.
If used in the routine, actually, it's also something that we need to work on. So what do you think about optical genome mapping or other technique like that?
Yeah, so there's been a range of different really great technologies that I think we could use to address this. And one is, as Bertrand Nadal points out, optical genome mapping. I think I'm very excited about this technology. And from a research point of view, I think it's really fantastic. The only drawback is, as far as I know, and he may have additional information, it really requires high quality DNA, and that's not possible, as far as I know, on formalin fixed paraffin embedded material. So I think that if you've got a center that's doing a lot of work with fresh frozen material, or you can get an FNA, go back and get an FNA or something like that after having established the diagnosis for this purpose, I think it's a fantastic technology. I'm just not absolutely sure where it belongs, where the currency really is formalin fixed paraffin embedded tissue. The other technology that I'm conflicted on, I haven't had a lot to do with it, but it is in my conflict of interest statement, is using high C on formalin fixed paraffin embedded tissue. And there are assays that are being commercialized looking at high C, where they'll identify a range of different rearrangements, but also can identify whether enhancers are engaging with MYC, for example, which would tell you a little bit more about the functionality. And from a research point of view, I'm excited about those because I think that they may answer questions such as, why do you have double hits that don't have the dark zone signature? Is this going into space where you don't have an enhancer, or is it somehow separated from its enhancer, to explain those findings?